All right, so the final topic in the responding to antigens part of the study design is looking at the adaptive or specific immune response. And you can see there's a few bits in this that we need to cover, but looking at B lymphocytes, antibodies, and also T cells or T lymphocytes. If we go back to our flowchart here, we've covered everything on this side, and now we are looking at this, our specific immune response. So the adaptive immune response is also known as the third line of defense, the specific immune response, and this is really looking at the fact that it's a specific response for specific antigens. So our innate response was just general responses to kind of any pathogens in the body, things like inflammation, fever, whereas now we are looking at specific responses. A really important feature of this response is that it gives us memory of specific pathogens, which means that in the future, if your body encounters the same pathogen again, we get a faster response. And this is what we often refer to as immunity. So being immune to a disease means that your body has generally encountered it in some way before it's encountered that antigen and is able to launch a faster response. There's two kind of main types of cells involved in this. We've got T cells and B cells, and they are properly known as lymphocytes because they are cells that live in lymphoid tissue. So T cells or T lymphocytes mature in the thymus gland. So remember, all lymphocytes originate in bone marrow. These ones move to the thymus gland and mature there. They have specific receptors for specific antigens, and we have these three different types, which we'll go through in a little bit. T cells, you'll see that helper T cells play a role in a few different responses, but in general, T cells are mainly involved in what we call this cell-mediated response. And this is for targeting infected cells. B cells or B lymphocytes originate in bone marrow and then they stay there. So that's where the B comes from. They mature in the bone marrow. They produce these proteins called antibodies, and we'll look at those in a bit more detail. And we have two types of B lymphocytes. We've got plasma cells and memory cells. And these are generally involved in what we call the humoral response, which again, we're going to go through in a lot more detail. So the initiation of the adaptive response is basically how this response begins. So remember we looked at in inflammatory responses, we get phagocytes called antigen-presenting cells, which can be macrophages and dendritic cells. After they have destroyed or kind of gobbled up a pathogen, they display the antigen fragments of that pathogen on their MHC2 marker, and they travel to the closest lymph node. Okay, so we looked at that in the lymphatic system. When the antigen presenting cell encounters a helper T cell that has a specific receptor that is the same as the antigen it's carrying, they will bind together. And what we get is this antigen presenting cell here basically presenting this antigen. If it matches the receptor on this helper T cell, this helper T cell gets activated. That helper T cell will then coordinate or initiate one of those two responses that we mentioned before. So it might kick off the cell-mediated response, which is for intracellular pathogens, pathogens that are inside the cells. This is usually viruses. There are some intracellular bacteria and parasites, but usually we're talking about viruses here. And it's also the response for cancer-infected cells. The other response is the humoral response. So helper T cells are going to initiate this response for extracellular pathogens, pathogens that are kind of floating in the body but aren't inside cells. And so this could be bacteria, it could be viruses as well, or parasites. And again, we'll kind of look at that in a bit more detail further on. This is just a kind of summary diagram that shows these two responses. Just note they both start with a helper T cell being activated by an antigen presenting cell. That's always step one, whether it's the humoral response or the cell-mediated response. So we get this helper T cell gets activated. It may then activate this cell response, or it may activate this humoral response. We'll start by looking at the cell-mediated response. So once that helper T cell has been activated, it will release cytokines, signaling molecules. And those cytokines do a few different things. One thing that they'll do, and the cytokines are kind of these little red dots in this diagram, one thing it will do is stimulate more helper T cells, because we're going to need more of these cells to be produced. So they'll start to go through mitosis and stimulate more helper T cells. They'll also activate our cytotoxic T cells. Okay, so that's what the TC is, cytotoxic or killer T cells, again with that same specific receptor. Those cytotoxic T cells will then differentiate into either active cytotoxic T cells. So these are the cells that are basically going to travel around the body and destroy the infected cells. So they're going to fight the infection. So most of the cytotoxic T cells will become these active cells to fight the infection. But also some will differentiate into memory T cells. Their job is basically to stay in the body, and then in the future, if we encounter the same pathogen, let's say the same virus comes into our body again, they can launch a much faster response the second time. Just going to look quickly at the action of those cytotoxic T cells. So the way they actually destroy infected body cells. We've got the cytotoxic T cell here, and then we've got here our infected cell, and you can see, you know, we've got viruses in there, and we've got these bits of antigen that actually get displayed on the MHC1 marker of those infected cells. Inside these cytotoxic T cells, you can see we've got these things called perforins. Okay, so these are signaling molecules within the cytotoxic T cell. Those perforins will actually leave the cytotoxic T cell, and they will make holes in the membrane of the infected cell. And I'm sure, as you can imagine, if we make enough holes in the membrane of that cell will lyse. So basically, it will kind of open up, explode, and all those viral particles in there will get released from the cell. Okay, so it's basically stopped that cell from manufacturing new viruses, but also it's releasing those viruses back into the space outside of the cells. We'll come back to what happens with those in a little bit. So that's the cell-mediated response. We get our cytotoxic T cells. Most of them will become these active T cells. They'll travel around and they will cause lysis of infected cells, and some memory T cells will stay behind. We'll look now at the humoral response. So again, we've got our activated helper T cells. They will release cytokines that will activate B cells with specific antibodies that are the same as that antigen or complementary to the antigen. So where T cells have receptors, B cells have antibodies. We're going to look at those in more detail. When a helper T cell finds a B cell with the same antigen that matches, the same antibody that matches that antigen, it will select that cell. It's just a specific term we use. That B cell has been selected. Then what we're going to need is more of those B cells. Okay, we've got the right B cell with the right antibodies, so we want more of them. So those B cells will begin to divide. So those cytokines will send messages for the B cell to divide, and we say that that cell is being cloned. This entire process is called clonal selection. Okay, so we select the correct B cell with the antibodies that we want, and then it gets cloned. Okay, so clonal selection is a specific term, and it does quite often come up in the VCE exam on this topic. Again, those activated B cells that have now cloned will differentiate into either plasma B cells. These are kind of our fighting cells. They're going to release the antibodies that will travel around the body and target the pathogens, or they may become memory B cells. Again, this will just be a smaller number of those activated B cells. Again, similar to memory T cells, they'll stay in the body, and next time the same pathogen is encountered, they can launch a much faster response. This is just a bit of a diagram showing that clonal selection. So we have our antigen presenting cell here. It has traveled to a lymph node. It has presented the antigen on its MHC2 marker to a T helper cell with a specific receptor. Okay, so it's activated that T helper cell. The T helper cell now will basically look for a B cell. So these are all just examples of B cells. It's going to look for a B cell that has an antibody that matches this antigen. Okay, so if you look on here, that shape doesn't really match the antigen, so that won't be selected. That doesn't match the antigen. This doesn't match the antigen. This here matches that antigen. Okay, so that is the B cell that gets selected. Okay, it's the one with the correct antibody. So the helper T cell will travel over here. It will bind with that B cell, and then we will get our cloning occurring here. Most of those cells will become our plasma B cells. They're going to produce these antibodies. And then, of course, we'll have some become memory B cells, and they will remain in the body long after the infection has been warded off. Okay, so that's just showing that again. Don't want this one, don't want this one. We've selected this B cell because it has the correct antibody for that antigen, and then we have our cloning down the bottom here. So we need to talk a bit more about plasma B cells, and particularly these antibodies and how they work. So plasma B cells produce very large numbers of antibodies, and this is a very fun fact here. B cells, so one B cell can secrete 10,000 antibodies per cell per second. Okay, this process of the selection and then cloning and release of antibodies can take several days, which is generally why, you know, you might feel sick for a few days before you get better. It takes a bit of time for your body to produce this response. If you look over here, these little pink dots are, you know, examples of my plasma B cells. Remember, each one of these is producing up to 10,000 antibodies. These antibodies get released. Okay, so they actually get released and travel away from the B cell, and they will travel in the bloodstream to the site of infection, or wherever those pathogens are in the body. Okay, when these antibodies find pathogens, and I've got two examples here. We've got one here which is a bacterial cell, and then this is a virus. Okay, when they find those pathogens, they will bind to the antigens. So the antigen site, which we'll go through in a second, will bind to the antigens on the surface of the pathogen. There's a few different things that they will do to basically destroy that pathogen. If it's a virus, remember, viruses need to get inside our cells to reproduce. So if an antibody binds to the surfaces of a virus like this, that can prevent that virus from entering our cells. Okay, so a virus that can't reproduce is quite useless. Okay, so by binding, these are the antibodies here, by binding to the virus surface, that virus won't be able to enter the cells and reproduce. Another thing they might do is tag the pathogen for cytotoxic T cells to come and destroy. Okay, so remember, our cytotoxic T cells release perforin to destroy infected cells. They can also do that to bacterial cells and cause, you know, holes to form in that bacterial cell wall and cell membrane and destroy that cell. Another thing that antibodies can do is attract phagocytes to that area so they can engulf the pathogen. And that would then go through the same steps of phagocytosis that we have looked at before. Okay, so a few different ways that antibodies neutralize pathogens when they find them. Just looking a bit closer now at the antibodies themselves, and this is something that comes up quite a lot in the VCE exam. There have been questions in the past where students have been asked to draw an antibody, or at least to kind of recognize the components. So antibodies are Y-shaped proteins, and as we've mentioned, they're produced by B lymphocytes. There are 5 different types of antibodies. You don't generally need to know too much about these, but you'll see that one particular type of antibody comes up again when we look at the allergic response. But these are the 5 different types: IgG, A, M, E, and D. If you look at these, and we're going to go through the structure a little bit in a second, but each antibody actually consists of 4 different polypeptide chains. Okay, so they are a really good example of a quaternary protein structure. And like we said before, the antibodies get released from B lymphocytes and then travel in the blood to the site of infection. So if this is my B lymphocyte here, you can see all these antibodies, the little blue bits all over the surface, but then they actually get released from that B cell. They'll find the closest bloodstream, enter there, and then they can travel to wherever they need to go to fight the infection. Okay. If you went on Google now and looked up antibody structure, you're going to see lots and lots and lots of different ways of representing it, but there's always the same kind of key things, which we will look at here. This might look a little bit complex, but if we break it down a little bit, antibodies, like I said before, have 4 polypeptide chains. Okay, so if you're going to draw one, you can start by drawing a big Y, okay, which is this bit here. That big Y consists of what we call heavy chains. They're the longer chains. Okay, then on the outside, just in this top bit, we have these 2 smaller light chains. Okay, so the light chains are those smaller ones. They're always on the outside. And then each of the chains is joined together or held together by disulfide bridges. Okay, so they're just bonds that hold those chains together. The 2 top bits of the Y here are what we call the antigen binding sites. Okay, so these are basically the specific complementary shape to certain antigens. So if this is 6 different types of antigens, that doesn't fit with that shape. Neither does that, neither does that, neither does that, neither does that. This is the antigen that is complementary to this specific antibody. If you had to draw this in an exam, you wouldn't need to put in that much detail. You could certainly draw it like I've done there, but just knowing that this top bit is the antigen binding site. Now, because we've got this top bit that is different for every antibody, we refer to this as the variable region. Okay, so this section here of the antibody is variable, meaning antibodies for tetanus and antibodies for chickenpox will have a different variable region, because that's the bit where the antigen binding site has to be specific. The rest of the antibody is our constant region. It's the same for all antibodies. Okay, all antibodies are the same except for that top variable region. Okay, so you should definitely pause here and make sure you've drawn and labelled an antibody with your light chain, heavy chains, and that antigen binding site. A lot of students get a bit confused with which response happens when. So I'm just going to try and cover that again. So which response is it the cell-mediated or the humoral response for certain types of pathogens? Okay, so we'll start with intracellular pathogens, for example, viruses. If we look here, this is a cell. We've got a cell here. Then we're going to have lots of space between cells. We're going to have fluid between those cells. But viruses, as we know, can be outside of the cell. They can be, in the blood plasma. They can be in the fluid between cells, but they generally go into the cells. That's how they will reproduce. Okay, so we've got viruses inside cells here and also some outside the cells. So if we look at this picture, we've got cells here. So let's say we've got lots of little bits of virus in here. But then we also have the viral particles outside here. We've got some virus in the blood plasma. Okay, so there's viruses inside cells and outside cells. So we know that our cell-mediated response will target the viruses that are inside cells. Okay, so we might get cytotoxic T cells traveling to these cells and releasing perforin that will basically destroy these cells. We'll get them to lyse, and those virus particles will then be released out into the space around. Okay, so we know we have that cell-mediated response. But we still need to get rid of these viruses, don't we? The virus particles that are outside the cells or in the blood plasma, they need to be destroyed. So then we will also have the humoral response to target those pathogens that are outside of the cells. Okay, so we'll also have our plasma B cells, and they have released their antibodies that now travel around. They might come through the blood, and as they're coming along, they will bind to these viruses and mark them either for destruction or stop them entering other cells. Okay, so we have a cell-mediated response and a humoral response working together. Keeping in mind, when those pathogens are destroyed, our B memory cells and our T memory cells will stay in the body so they can do this quicker next time. For extracellular pathogens, and we're often talking about things like bacteria or fungi or parasites, this is when the pathogens are in the body tissue or the bloodstream, but they're not inside the cells. So if we look here, that pink area is our tissue, that's our cells, and then we've got all these bacteria kind of growing around the cells. Okay, they're too big to fit into the cells, but they're crowding that tissue and they're causing lots of problems, and they might be releasing toxins that are slowly killing the cells there. Okay, so they're crowded around, but they're not in the cells. Same thing here in the bloodstream. These blue things are the bacteria. They're in our fluid, but they're not inside the red blood cells. They're not in our body tissue. They're in the bloodstream. So they're extracellular. Go back to this picture now. We'd be looking at things like this. Okay, so we might have bacteria growing all around our cells, and you can probably see that that's going to cause lots of problems as they divide and take up lots of space. They might enter the bloodstream as well. Okay. There's no bacteria or there's no pathogens inside the cells at this point, so we don't have a cell-mediated response. There's no point destroying this healthy cell because there's nothing in there. So in this example, we just have that humoral response. Okay, so we've got our B cells release our antibodies, and they will travel and they're going to bind with this pathogen, and then they will either attract cytotoxic T cells to come and destroy it, or attract some phagocytes to come and destroy it. Okay, so extracellular pathogens, we're really just getting that humoral response. The final thing to kind of talk about here is what we call our primary and secondary immune responses. The primary response happens the first time you encounter a specific pathogen. I want you to think for a second how you might encounter a pathogen, or I guess what we really should be saying is encountering an antigen, which usually is found on the surface of a pathogen. Obviously, the main way that this might happen is by getting sick. okay, by getting infected with that pathogen for the first time. Okay. Another way that this happens is through vaccination. We're going to look at that more in the next lesson. But anytime an antigen enters your body for the first time, we have our primary response. As I mentioned before, it takes a few days for antibody numbers to increase because it takes a few days for those B cells to get selected and then to be cloned and then to produce and release all of those antibodies. Important thing to note here is that we don't have memory cells yet because we haven't encountered this pathogen before. Okay, so basically if we were to look at antibody numbers, I've got a much better picture than this on the next slide. Antibody numbers and time. Let's say we get infected here. Takes a few days, but those antibody numbers start to increase as we're getting B cells being produced and then releasing antibodies. And then once we're better, those B plasma cells aren't needed anymore. So they'll go through apoptosis. All the antibodies have kind of been used up. So those numbers will drop right down. Okay. We will still have some antibodies on our memory B cells at the end of a primary response. Okay. The secondary response is the second or third or fourth or fifth time you encounter the same pathogen. Okay, so this could be that you get reinfected by something. Okay. Often with a lot of diseases, if you get it once, you might not even know if you get it again because your body reacts to it much quicker that you don't actually feel symptoms or you don't get sick. This is the same thing with vaccination. If you've been vaccinated and you've done this primary response, then you're not going to know if your body encounters that pathogen again because your body fights it off. This time, memory B cells exist. Okay, we've had a primary response first, so we have the memory B cells with those specific antibodies ready to go. Okay, so what that means is we will get a much quicker response and a lot more antibodies will be produced and released after that secondary response. Okay, and the numbers will actually stay higher of antibodies after that infection. This is showing that again. So we've got the antibody production over here, we've got time, we've got here our kind of first infection, so whether that's through getting sick or being vaccinated. We've got a few days because we haven't got any memory cells yet, and then we get our primary response. Okay, so plasma B cells are selected and cloned and they produce antibodies that fight off that infection. Over here, we're kind of recovering. We've gotten rid of the pathogen, so those numbers go back down again. Okay. Then in future, you get sick again, and this time we've got memory B cells. It doesn't take several days like it did over here. Those memory B cells get activated straight away, and they differentiate into plasma B cells, and they start producing antibodies really, really, really quickly. And you'll see we get much more antibodies that second time as well, okay, because those cells are ready to go. So we get a much faster, quicker response that will kind of fight off the infection. Okay, it's a similar thing with booster shots. When you probably remember with some of your vaccinations, you've had to get a booster shot maybe after a year or so. Same kind of principle. We're getting this increased antibody response. But again, we'll look at that when we look at vaccinations. Okay, so we've looked at the components of the adaptive or specific immune response, including our B lymphocytes and their antibodies, and that humoral response, as well as the actions of our T helper and T cytotoxic cells and the cell-mediated response. All right, that's it.